Have you ever noticed how plants and animals seem to know when the seasons are changing? In places with noticeable seasons, like summer, winter, spring, and autumn, things like temperature, sunlight, and even food supply can change a lot. Even in tropical regions, the environment shifts, usually with changes in rainfall. To deal with these changes, living things use something called photoperiod responses. This means they respond to the length of day and night. These natural responses help them do important things at the right time of year. For example, plants might grow flowers or form bulbs, while animals might hibernate, migrate, or go through a pause in development called diapause. These seasonal changes are closely linked to internal body clocks that help organisms prepare for what's coming next in their environment. In this video, we focus on photoperiodism in plants, the regulation of seasonal activities in response to day length, or photoperiod. The idea that plants can sense the time of year was discovered in the 1920s by two American plant breeders, Garner and Allard. They were investigating a new hybrid variety of tobacco that behaved strangely. It wouldn't flower outdoors, even after growing to 2 or 3 meters tall. But small plants of the same variety flowered easily in winter when kept warm inside a glass house. After testing different variables, like temperature, light intensity, and more, Garner and Allard concluded that day length was the key factor triggering flowering. They found that this particular tobacco hybrid needed short days to begin flowering, but the cold winter temperatures outside prevented flowering. They coined the term photoperiodism to describe the control of flowering by day length. Photoperiodism is the biological response of organisms, especially plants and animals, to the relative lengths of daylight and darkness in their environment. It's a natural mechanism that helps them adapt to seasonal changes by adjusting growth, development, and reproduction based on how long the day or night is. A clear example of photoperiodism is flowering in plants. In this process, the critical day length refers to the specific duration of daylight needed to trigger a biological response, such as flowering. This critical day length differs among plant species and determines whether they are classified as short day, long day, or day-neutral plants. To identify a plant's critical day length, scientists expose it to different photoperiods, or simply different day lengths, and observe whether or not it flowers. Short day plants flower when the day length is shorter than their critical day length. This means they need long nights and short days to bloom. For example, chrysanthemums typically flower in winter when there are around 10 hours of daylight and 14 hours of darkness. Long day plants flower when the day length is longer than their critical day length, meaning they need longer days and shorter nights. These plants usually flower in summer. An example is the sunflower, which might require 14 hours of daylight and only 10 hours of darkness to begin flowering. Day-neutral plants do not rely on day length to flower. They can bloom any time of year, as long as other conditions, like temperature and nutrients, are suitable. A common example is the tomato plant. However, it was later discovered that it's actually the length of the night, not the day, that controls flowering in plants. The key factor is the period of uninterrupted darkness, known as the critical night length. In other words, what truly regulates flowering is how long the night is. This means that long-day plants are more accurately described as short-night plants, and short-day plants are actually long-night plants. Garner and Allard's original classification of plants based on day length was later refined, thanks to two important types of experiments. 1. Light interruptions, when a long night is interrupted by just a few minutes of light. Short-day plants fail to flower, while long-day plants are triggered to flower. In contrast, interrupting the day with a period of darkness does not affect flowering. This shows that it's the continuity of darkness, not just the total amount of daylight, that matters most. 2. Non-24-hour cycles. Experiments with cocklebur, a short-day plant, showed that it did not flower when exposed to cycles of 4 hours of light, followed by 8 hours of darkness, even though 4 hours of light is much shorter than its critical day length. However, when subjected to cycles of 16 hours of light, followed by 32 hours of darkness, the plant did flower, despite the long day length. This further confirms that the length of uninterrupted darkness, not the length of daylight, is what controls flowering in these plants. These experiments confirm that the length of the night, not the day, is the key factor in triggering flowering. So, what we call short-day plants are really long-night plants, and long-day plants are actually short-night plants. But how do plants detect this light and dark signal? By exposing different parts of both short-day and long-day plants to varying photoperiods. Researchers discovered that the leaves are responsible for detecting the photoperiod, not the stem apex, where flowers eventually form. This suggests that once the leaves detect the correct night length, they send a signal to the growing tip, or apex, to begin flowering. In further experiments, scientists grafted closely related short-day and long-day plants together. Interestingly, regardless of the photoperiod, both plants were triggered to flower. This showed that the flowering signal is the same in both plant types. Despite many years of research, scientists have not been able to identify a specific flowering hormone, although it was given the name origin. What we do know is that some kind of chemical signal is involved. This signal moves through living tissue in a process that requires energy and is sensitive to temperature. Thanks for watching. In the next video, we'll explore the fascinating role of phytochromes in photoperiodism. Until then, happy learning.